what I've been asked to talk to you about today is sort of a general overview of stem cells and their potential for regenerative medicine and obviously precision medicine. So what is regenerative medicine? And it really comes in all shapes and forms. Really, it's all about restoring normal tissue or tissue function by basically repairing or replacing cells that have been damaged. And this includes, really, and this is where precision medicine comes into it, a better understanding of the causes and the progression of the different diseases, such as Parkinson's and diabetes and a lot of these genetic diseases. And like I said, this is where really precision medicine comes in, because this helps us, and also the studies that we're doing to study the evolution of these diseases. Regenerative medicine also involves creating a lot of new tools and technologies to basically accelerate the research and do drug discovery, and also medical testing. And this is where stem cells come in, because we can do this, a lot of this with stem cells, and this would really reduce the cost and also reduce the using animals as a model. Basically, we can use stem cells as a surrogate for the human patient to test these drugs on. And finally, we want to design and develop novel treatments that will basically either enhance the natural repair processes, so basically anything that normally happens but doesn't happen good enough, so basically to enhance that, or to replace damaged or diseased cells. And I think that's a lot further away as far as cell replacement therapies go, but we'll talk about that as well. So this is just sort of a schematic of where we want to go with this and how precision medicine is involved in this. So obviously we have a person that may have a degenerative disease, and as I'll talk about, we can derive cells from these patients and make them more pluripotent. And so we have these disease-specific stem cells that then we can use to basically model the disease in a dish, and this is actually becoming very effective. So basically, you can figure out what's actually wrong with those cells and do a lot with them in a tissue culture dish. Then we can use these cells to screen drugs. And I think a really common problem in my field's diabetes, and basically you have drugs that work on one patient and not another, and so you could actually use these cells to see. So if you're trying to make insulin-producing beta cell function better, you could take these patient-derived cells, make them into a beta cell, and then use drugs and say, does this drug work on this cell versus another cell? And also, this is, again, where precision medicine comes in. We can use these cells for sequencing to understand what the genetic defects are. The idea is then to identify the defects and replace them or repair them with a normal, a normal gene. And this has become really a lot more powerful just in the last year with these gene editing technologies that we're not going to go into today. But if anybody wants to know about them, we can talk about them later. And once we can correct these cells, you might be able to make a new cell that can be either reintroduced into the patient, but like I said, I think that's quite far away. So what are the types of health problems that can be treated by stem cells? And this is just a subset, but these are all the ones that are sort of on the radar right now and being looked at is neurological disorders. And actually, it's funny they don't include it here. Eye disorders are also, I think, one of the most advanced uses of stem cells is in treating eye diseases, limb amputations, spinal cord injuries. There's a lot of research trying to use stem cells to regenerate spinal cord neurons. Burn patients, so we need to get new skin. So again, this has been a very big avenue of research. Heart disease and diabetes. And this is just a subset, but these are all the different diseases that people are trying to say, okay, can we harness stem cells and the potential of stem cells to treat these different diseases. So this is just an outline of what we're going to talk about today, and like I said, if it's too basic, we can move forward, but I think we need to really define what is a stem cell. I think this is a problem. We use the term stem cell loosely as biologists, and I think it's important to understand what a stem cell is. I'm going to talk to you about where they actually come from and the different types of stem cells, because I think, again, the perception, if you're not working in the field, there's a lot of loose jargon that's used, and we really need to define what we're meaning by an embryonic stem cell versus adult stem cells. And then some of the technologies that are used to either derive new stem cells or to do gene replacement techniques. And then just at the very end, talk about the therapeutic ramifications of this. And please, any questions on the way, just interrupt me, that's fine. So what is a stem cell? Here we show a stem cell, and it can become a differentiated cell, and it also can produce more stem cells. And so there are really two hallmarks that make a stem cell a stem cell. But to be a stem cell, you have to be able to self-renew. So you have to replace yourself. And so a lot of people say progenitor cells, so cells that can produce other cell types are stem cells. But if a cell cannot self-renew, if a cell can't replace itself, it cannot or should not be categorized as a stem cell. And so that's the main feature of a stem cell. And the other feature is that it has to be, have the capacity to give rise to different lineages of cell types. And this can be as few as 2, but it's got to be more than 1. So it's got to give rise to itself and some other cell type. Okay? And this is where the literature gets really messed up. And so as far as where I work, again, in the pancreas, so people have looked for a long time for a pancreatic stem cell. So a cell that lives and self-renews and gives rise to other cell types. And so far, it hasn't been found. But a lot of people say progenitor cells are stem cells. And they're really not, because they don't self-renew. There are several types of stem cells, and we're going into this a little more deeply in the next few slides. But I guess you would call it, and I'll talk more about this, the prototypical stem cell is the embryonic stem cell. So this is a cell that is pluripotent, and I'll show you it's not totipotent, it is pluripotent. And it exists really only at the earliest stages of embryonic development in the animal. And it doesn't exist till after about 5 days of development in humans. You can no longer derive the embryonic stem cells. But you can take these cells and isolate them from the embryo and grow them in a dish almost indefinitely. And this is where the power of embryonic stem cells has been in a lot of the regenerative medicine techniques. So these are embryonic stem cells, and these are also what you hear referred to as ES cells. The adult stem cell is much different. So it doesn't have as much of a capacity to become other cell types. So we refer to that as being multipotent. And these are generally tissue-specific stem cells. And examples of these are like your intestinal stem cells, or your skin stem cells, or your bone marrow cells. And these cells are normally in the body of the adult, and they have a more limited potential than an embryonic stem cell. They usually just give rise to specific lineages, and I'll show you a couple examples of these. And they can't be grown indefinitely. So the embryonic stem cells can be passaged over and over again. The adult stem cells have much more limited capacity. And then the third type of stem cell we're going to talk about is the induced pluripotent stem cell, and I'll tell you how these were derived. And these are cells that are derived from a somatic cell that can be taken from any tissue, but for the purposes of regenerative medicine and precision medicine, it's usually derived from the skin or the blood, and this is just because it's easy to take a scraping of skin or to get blood from a person rather than going and taking a tissue that's internal. And again, these cells are pluripotent, and they actually have a lot of the characteristics of embryonic stem cells. Okay, so these are the 3 major types of stem cells we're going to talk about today. There's also this idea that there is a cancer stem cell out there, and these are supposedly are thought to be the cell of origin of the cancer. And it's a very small part of the tumor, and these cells self-renew and give rise to other cell types within the tumor. And we're not going to go into that at all because it's just a whole other level of stem cell biology. Okay. So stem cells are not something we invented. They actually have a normal role in the body. I think sometimes we forget about that. So obviously during embryogenesis, during fetal development, all of the tissue is derived from a stem cell, so it's for the generation of fetal tissue. There are stem cells, as I said, in the adult that are normally there to renew adult tissues for normal turnover. Good examples of this are the hematopoietic system, your hair. You're constantly regenerating your hair throughout your lifetime. Your skin, you're constantly renewing your skin, and the small intestine. So these are all known and well-characterized endogenous adult stem cell populations. There's also populations of stem cells that are basically there to give rise to damaged cells. And again, the liver and the skin are a good example of this. So just to give you an example, here is the intestine. In the mouse and the human and in all the organisms that have an intestine, there's a constant renewal or turnover of your intestinal cells during your lifetime. Actually, every 3 to 4 days, the cells of your intestine turn over. So they're constantly renewing. And it's really well characterized now. There's 2 stem cell populations within the intestine. There's this stem cell population that's down here. These are these red cells that are the most common stem cell population, and they give rise to all the cells. And so as these cells, they migrate up, they actually start to proliferate here, and then they start to differentiate into the different cell types of the intestine. So this one blue cell, in a course of 3 or 4 days, will start to migrate, differentiate, and at the very end will basically be sloughed off, and this goes on throughout the lifetime. There's also a second stem cell population. Let's see where they put those here. I can't even see them on this. It's supposed to be the +4 cell. I guess it must be this green cell here. And again, so there's 2 populations in the intestine. One is your normal, natural population, and the other one actually, in times of injury, gets activated to take over for that population. So this is one of those tissues, and it's similar in the skin, like I said, in the hair, that you normally have a stem cell population in your body that's constantly renewing. The question is, can we learn anything from these cells for the other tissues that maybe don't have stem cells or maybe have really rare stem cells? And that's why the intestine has been studied so well. So really the goal of stem cell research has been, in the more recent years, is to basically harness existing stem cells or create stem cells so that we can basically transplant them into, say, a damaged area and regenerate that area so they can replace them. This has been, obviously, with bone marrow transplants, has worked for a long time. But say for skin, for burn patients, can you evolve a stem cell population that you can use to treat burn patients? There's also this idea that, you know, if there is a stem cell population, there's often in many tissues what we call facultative stem cell populations. So they're sort of inert populations that upon injury can be activated to replace cells. And so the idea is, okay, if injury can do this, can we also do this? Can we actually take a cell population that can be induced to be a stem cell population or activated? Can we figure out how to activate that population? And this is now, I think, and so these 2 areas of research are ongoing, and I think a lot of progress has been made, but I don't think we're there yet. I think where the most immediate potential is to use stem cell research as I said, developing models for diseases or drug discovery, and this is where they've been really, really made a lot of progress. Okay. So embryonic stem cells. So everyone's quiet. You can always interrupt. Okay, embryonic stem cells is, like I said, the prototypic stem cell. So this is a stem cell population in the embryo that can give rise to every cell of your body. They are, as I said, immortal. They can proliferate indefinitely, and they make all cell types of the adult body. And so the way we know this is just showing an example in mice, but we can take, and here is actually a very early embryo, and it's the blastocyst stage of the embryo. And what you can do is you can see these little cells here. These are embryonic stem cells that we put a red fluorescent marker in it. This is a cherry marker. So we now you're physically injecting these cells into the embryo. They become incorporated into the embryo, and you can get a mouse that now is glowing red. So this is how it shows us that this cell, this stem cell, can give rise to every cell within this embryo. And then these mice are derived from that cell. Okay, so this is one of the first examples in mice that this could be done. Just to show you the stages where we can get these embryonic stem cells from, this is just showing the first week in human development. So you have fertilization. The fertilized egg starts to divide and differentiate. So you have the 2-cell stage, 4-cell. And it's at 5 days after fertilization is the stage where we derive embryonic stem cells from. I think I took it out. I had a slide that showed that this was basically the same size as the eye on a dime. So it's very small. But this is the stage we get. And this is just showing another picture of it. So again, you've got the 2-cell stage, 4-cell, and now you've got what I said was the blastocyst stage. And this region here is the region that's going to become the embryo proper. And this is where we can actually derive these stem cells from. And we call this the inner cell mass. And this is just a cartoon of that. So these are the cells of the inner cell mass that will normally give rise to the embryo. And so this is where we derive these embryonic stem cells from for research. And again, the one thing is that at this point now it's been worked out as a really well worked out technique. They're easy to isolate. You take these inner cell mass and you can plate them in medium. But the problem ethically is that the embryo gets destroyed in the process. So this is where people have problems with embryonic stem cells. But there are sources. Most of the sources in humans come from a surplus of embryos from IVF. You can actually use in vitro eggs and sperm. You can basically do this in a dish. And also germ cells from aborted fetuses. So these are the 3 major sources of human embryonic stem cells. But what I should say is maybe I'll go maybe I'll go through the process first, but then basically once a stem cell has been made, and the human ES cells that we use in research, probably less than 100, but there's some that are, once they're made, people just can reuse them. So we don't have to constantly get new stem cells. So, and there's an approved list by the NIH that everyone uses, and they all have some different features, but this is not something every time you want an embryonic stem cell or every time you want to do embryonic stem cell research, you don't need to go back to the embryo. You can just take from the freezer, you can order it from the NIH, you can get these different cells already made for you. But just to show you how they're made, it's not like, it actually turns out to be very simple. So what people do is they isolate these cells. I'll go back one. You isolate these cells here, these green cells, and you basically plate them onto a layer of fibroblast cells, which provide all the growth factors to allow these cells to live. And you culture them for several days, and then you basically just disaggregate them and reaggregate them. And what you get, and there's no selection, but what you get is you get colonies. They look very different, but you get colonies that are differentiated and colonies that are undifferentiated. So colonies that still have stem cell potential. And you basically just hand-pick these undifferentiated colonies and then replate them. And you do this several times until you have a pure population of cells that remain undifferentiated. And it's all done by looking at their morphology. And so it's basically just a technique where you're plating cells, replating cells, and just selecting for those that retain their pluripotent status. And so once you have that cell line, that cell line, like I said, can be maintained forever in culture. There's some features you really want to make sure, because as you can imagine, you're taking these cells out of an embryo and you're putting them in a completely artificial situation. So you have to make sure that they maintain their diploid karyotype, so they maintain their normal number of chromosomes. They can still colonize fetal lineages, and so this is important for mouse work when you want to make genetically engineered mice. Like I said, this is not something we're probably going to go into humans with anytime soon. But for mice, that's what we really need, is to make, say, our knockout mice or our transgenic mice. They're pluripotent, and I'll show you this in a second, because you can't just take a stem cell and make a person or a mouse in a dish. and I'll show you why in a second. But this is really because they don't give rise, the stem cells only give rise to the embryo, the cells of the embryo, and not any of the extra embryonic tissue, and not any of the other cells that are needed to promote survival of that embryo. I don't know if it's in the next slide. So this just illustrates what I was saying. So basically, you take the cells out of this inner cell mass, and normally, if in vivo, in a mouse or in a person, these cells would give rise to all the lineages of a person. But when you take these out and you make them into embryonic stem cells, they actually only give rise to the cells of the embryo, so the mesoderm, the ectoderm, and the endoderm. And this is visually easy to see in this slide because here is a mouse embryo. And you can see it's not completely blue, but what we've done is we've knocked LAC-Z into these embryonic stem cells. So this is just a blue marker that you can see can give rise to all the cell types of the body. But you see this is the yolk sac, and none of it comes from that embryonic stem cell. So basically, you can only use a stem cell to give rise to the embryonic cells and not these lineages. In theory, you can propagate them forever, but as you can imagine, every time you propagate them, there's a chance that they'll lose a chromosome, that they'll get a mutation, that something will go wrong with them. And so what people usually do when they make their first embryonic stem cell, they freeze their first batch. And so you always have that first batch. And then you can pull it out. And so it's even, if anyone has done stem or even cell culture work, you constantly want to go to a low passage number because no matter what, the more you propagate them, there's a more chance you're going to have them become mutated and not work as well. Yeah, good question. So stem cells have this potential to give rise to every organ in the body in vivo. So when you take this stem cell, like I showed you, and you can put it back into the blastocyst of a mouse, it can give rise to every organ within that mouse. But can it give rise to all these different cell types in vitro, in a dish? So if you take a stem cell and you put it in a dish, you say, okay, now I want to make a liver cell, or now I want to make a bone cell. It turns out not to be as easy to do that as it is in vivo. So we don't know all the signals that it takes to get there. So basically, even though they have the capacity to produce every cell type in vivo, it's harder to get this to work in vitro in a dish. And this is where the majority of the research has been for the last 10 years, is trying to direct the differentiation of these stem cells into a certain cell type. And this is where precision medicine really, it needs this as a tool, because say you have, well, maybe I shouldn't get into that yet, but basically the whole idea, say for a replacement for type 1 diabetes. In type 1 diabetes, you lose your beta cells from an autoimmune attack. Say you want to take a stem cell and make more beta cells so that you can replace those beta cells that are gone. Can we do that in vitro? We can do it in vivo, but can we do that in vitro? And currently, we can't. We've gotten a lot better at doing this with a lot of different cell types. But we can't say, okay, I'm taking a stem cell and making cell type A versus cell type B. What we can do is we can say, I'm going to make a stem cell, and we can get sort of a mixed population of different cells and then maybe select for the ones we want. And that's what people have started to learn, actually from studying mouse development mostly. So learning how the mouse develops and the signals and the regulatory factors that are necessary for mouse development, we then say, okay, well, if you need factor X in this stage of mouse development, maybe we can put that on these cells in a dish and it'll help. And that's really where it's been very, where we've learned a lot. And it turns out that the best way to make a specific cell type from a stem cell is to go through the same stepwise conditions that you actually go through to make a cell in the embryo. So this is basically an example. So say you want to take this embryonic stem cell and you want to make a liver, a lung, or a pancreas. Or if you want to make muscle or blood or skin. It turns out that you just can't take this cell and throw on a growth factor, and it'll become the cell type you want. What we've learned, you have to basically go through the same program. So if you want, we know that liver and lung and pancreas are derived from endodermal tissue. So if you really want to take a stem cell and in a dish make it into a liver cell, you actually have to first say, okay, now first I'm going to make you into an endodermal cell, and then I'm going to make you into a liver cell. So you can't take it and say, okay, I'm going to make a mesoderm cell and then try and push it in that direction. It turns out that these cells, I mean, nature's done it very, very precisely during development, and it turns out that you have to really recapitulate a lot of that in a dish. And there are a lot of early studies with stem cell research that didn't do this, and they got all these cells that really didn't resemble the cell type they were looking for. Again, because this is the field I'm in, this is where it's been really, really successful in this stepwise differentiation of cells. So what they want to make is insulin-producing beta cells. And so what they did is they go through a stepwise differentiation procedure that mimics what's happening in development. So normally in development you have a stem cell gives rise to an endoderm, gives rise to gut endoderm, gives rise to foregut endoderm, then gives rise to pancreatic endoderm. And then finally, at the end of the day, you get islets. So they've actually repeated all of these different conditions, but in a dish, and that's gotten them to an insulin-producing cell. So you can't go from here directly to here. You have to go in this stepwise fashion, and at the end, you get a cell that resembles a normal cell. This is now how it works for a lot of different cell types. The problem is that we still have inefficient differentiation. So this looks great, but if it turns out that only 1% of these cells in a whole dish are the cell type you want, then it's not really that useful. So right now, it's still very inefficient. Also, it turns out for almost all of the different, like the pancreas, the nervous system, for a lot of these different tissues, what we're having a problem with right now is the cells at the end of the day are still immature, and they still don't function properly. So they look more like a fetal neuron or a fetal beta cell. And so this is a hurdle that I think everyone in all the fields of stem cell biology are sort of stuck at this right now. And it seems like we make incremental progress, but there is progress being made. Also, like I said, you get a dish full of mixed populations, so at some point you're going to have to figure out how to sort out those different populations. And also, if you transplant these cells, if you've got a mixed population of some cells that are differentiated and some cells that are still not differentiated, you can get tumors formed. So again, there are some roadblocks for trying to get by, but it's even compared to last year, there's a significant amount of progress being made. So we're getting closer and closer and to be able to take an embryonic stem cell in a dish and turn it into a cell type of choice. And actually, this is again the beta cell, and this is just is it this year or last year? This is where they've gone from taking a cell that was not a beta cell that made insulin but wasn't responsive to glucose. They now have a cell that's responsive to glucose. So again, it's this, we're making advances. The other problem that we have when we think about therapeutics of stem cells is there's still an immune rejection. I mean, people say, well, stem cells don't initiate an immune response, but if you differentiate them into a cell type, they're going to express non-host surface markers, and they're going to induce an immune response. Like I said, we're still having some difficulty in directed differentiation. They can form tumors. And even if you have a stem cell, so say I can finally make an insulin-producing beta cell, and I'm going to replace the cells in my islet. How am I going to get it there? So is it going to be surgery where I actually put them there? So that's the other thing. How can these cells home to the right place? And also, how long do they survive? If you did replace them in a body, say you wanted to replace liver cells, how long are they going to be there and functional? So these are all things that are now, all of these, once we get the cell, we're still going to have a lot of work ahead of us to figure out how to use them. The embryonic stem cells has been where most of the research has been. We've used them in mice for a long time because it's taught us a lot about genetic engineering. But as I said, for humans, there's ethical considerations, and so they've been used a lot less. Adult stem cells are interesting in that they are a normal endogenous population. And like I said, there's hematopoietic stem cells, neural stem cells, skin stem cells, intestinal stem cells. And in about 5 years ago, maybe a little more, there was this real excitement that maybe these cells could be harnessed and to be turned into other cell types. I don't know where I'm going with this. So it once was thought that basically, say, an intestinal stem cell could only give rise to intestinal cell types. Or a bone marrow cell could only give rise to blood cells. But about 5 or 6 years ago, maybe even 10 years ago, it started to be appreciated that you might be able to direct these to other cell lineages. And there was this idea that maybe there was more plasticity. Maybe we could take a hematopoietic stem cell and direct it to be a pancreatic beta cell. And so there was a lot of research that went into that area. And this is just one example where it's actually, this is from Mimo Achille, who's actually here in the Berry Center. And he was actually able to take, well, this is, I should show you what this is. This is a gut, a section through an intestine, and you have blue is marking where the cells are. And none of these cells in the intestine normally make insulin, but here you've got a pancreas that makes insulin. And he was able to make a mutation that specifically allowed these intestinal stem cells to give rise to insulin-producing cells. So there is some plasticity that you can have with adult stem cells, but it turns out that it's not quite as good as we thought it was. And there was a lot of, I think, now that we have induced pluripotent stem cells, which I'll talk to you about in a sec, people are moving beyond this. But I just wanted to say that it is a possibility. So basically, the idea is that some stem cells from different sources maybe have more flexibility than we thought to become. But we're going to have to figure out how to manipulate them, and that's not been easy. Bone marrow cells would be great because we can get them out, and it would be great if they harbored, it's believed that they might harbor stem cells for different tissues. That, again, has not, that's been proposed to happen, and it's a hypothesis, but there's been no evidence that that's actually happening. And also, like I said, now the multipotency, the ability of these cells to be plastic is called into question because it turns out some of the earlier studies weren't, basically, these bone marrow cells were migrating to a different tissue, and instead of turning into the cell type, they were actually just providing a nice environment for endogenous stem cell to proliferate. So people have moved away from adult stem cells, but I thought I just wanted to show you a few slides on this because it might come up in some of the studies that you hear about in this lecture. So this is where I think we are right now as far as where all the research is being focused on, and also where I think precision medicine is definitely having an influence. And this is using nuclear transfer and induced pluripotent stem cells. In both these cases, the idea is that you can get patient-specific stem cells. So when we were talking about embryonic stem cells, I said we only have, 50 to 100 stem cells out there that have been made, but they're not obviously patient-specific. They were made from some source of embryonic tissue, and everyone uses them for research. But if you really wanted to start applying this to treating patients, the idea is to get patient-specific stem cells. And there are now two major ways of doing this. So basically, you take a biopsy from the patient, like I said, either the skin or you can get blood, and you can use a set of defined factors to make these induced pluripotent stem cells, and I'll talk about that second. Or you can actually, there's a way to take the nucleus out of a person's own adult stems, or adult, say, skin cell, and reprogram that cell by transferring the nucleus. And I'll show you that in a second. But if you can do these, then you get patient-specific stem cells, and this is where precision medicine is really going to benefit from this. And so, and then again, like I said, differentiate these cells into the different body types. So I think I'm going to talk about nuclear transfer first. You guys probably have already heard about this and may not have known it. So the Nobel Prize in 2012 went to John Gurdon, who was the, one of the first people to show that this could work. And you can see these are all the clones of the frogs he made. But you probably heard about it mostly when Dolly came around, or some of you might be too young, actually, for Dolly. But so basically, this sheep, Dolly the sheep, was cloned by nuclear transfer, and I'll show you how that works. So basically, what you do is you can take a cell from my skin, and you can take out the nucleus, and you can put that nucleus into a cell that has been derived from an oocyte, an egg from an embryo, and you take out that oocyte nucleus, you replace it with the donor adult cell nucleus, and it reprograms that cell. It's something in the cytoplasm of that cell that makes that nucleus be reprogrammed so it's more naive. So it's no longer a skin cell. It's now been reprogrammed to be a very pluripotent cell. And then you can take that cell, just like we took ES cells, and put them into an embryo, and you can actually make a lot of different cell types. And that's how they made Dolly, right? So they took an adult sheep cell, they put it into an embryonic, or nuclei of an embryonic sheep cell, and then they made this whole sheep. And so this was all, of course, will there be another you? Could we all have clones of ourselves? It turns out that this is very, very inefficient. So most clones actually die after implantation, so they had to try and do this with a lot of different sheep before they actually got one. It's very inefficient. It's costly if you wanted to use it for routine treatment. And the thing is, if you're doing this with humans, we go back to the same problem. This oocyte comes from an embryo, a human embryo. And so, again, there's ethical issues of using human eggs to make this therapy work. So this is, I think, losing a lot of it. There are still people working on it. In fact, here at the university as well, but it's become actually less of an option because of what I'll tell you in the next part, the induced pluripotent stem cells. Where it has been actually really useful is the very similar technologies have been used to actually replace defective mitochondria. So basically what they're doing is, say there is a woman that has a mitochondrial defect, what they've been able to do is remove her mitochondria and get her mitochondria from a healthy oocyte, and then basically that egg with the new mitochondria is fertilized. That means actually this one egg is derived from 3 different genetic sources. So mitochondria from one woman, in the egg from another woman, and then the sperm does the fertilization. So it's, and this is one way they are looking at treating mitochondrial disease. So this is actually being done in humans, but it's very specific for mitochondrial disease. Okay, so that was nuclear transfer. Like I said, that had its heyday, but now with the reprogramming that's been worked out, this is where the future is, I think. So this is, again, Shinya Yamanaka actually shared the Nobel Prize with John Gurdon, and he discovered the way to do make these induced pluripotent stem cells. And basically the way this is you take a somatic cell, again, a skin cell, a hair cell, any cell type, but anything that's basically an adult cell, and you can reprogram it just by giving it a set, and he found this set of 4 different factors. These are all transcription factors, but basically you can express these transcription factors in this cell, and you can make what they call an induced pluripotent cell, which is really equivalent to a stem cell. It is pluripotent. It can actually give rise to all the cell types of the body. Basically, these cells, you get the somatic cells, you add the reprogramming factors, and initially they did this with viruses. Obviously, when we're doing this for humans, we don't want to use viruses, and now people are deriving ways of adding these factors without viruses. You basically grow these cells in a culture condition that you would grow embryonic stem cells in, and over time, several of them become induced pluripotent cells. Now we know, and they did it for mice first, and they did it with retroviral infections, and then they did it, I guess this is still mice, and then now they've also done it for human cells. So this just shows what the potential of this is. So you can take a patient who has a disease, you can take a biopsy of their cells, you give it these 4 reprogramming factors, and you induce these cells to be pluripotent. And by pluripotent, they actually are very similar to embryonic stem cells. You can then take those cells, and if, say this person has, we'll say, hemophilia, so then basically you could basically correct that gene that's mutated and then make a blood cell and either study it in a dish, or the hope is eventually to transplant it, the cells that have been corrected. So this is essentially where precision medicine comes in, because you need to know the gene that needs to be corrected. And so the thing is, that this patient, we should know, okay, this person has a defect in this gene, so we can actually take their cells, induce pluripotency, correct that gene, and then make those cells better and put them back in. Now, like I said, I think we're really good at all of this. We're getting better at this. We're not doing this yet. But that's where the future is. Where I think that the immediate future is we're getting these mature cells, say a neuron or a beta cell. You can now grow these cells in a dish, either the mutant cells or the corrected cells, and this is where you can do therapeutics. This is where you can say, does this drug work on this cell? Does this drug treat this defect? And that way, you're not treating it on animals, which are unrelated. You're not treating it on the patients themselves. You're treating it on their cells. And so you could imagine you could test 96 different drugs at one time on the person's cells, where you could never do that in a drug trial on a person. So I think that's where the real benefit is. And again, you can do this in drug discovery too. Say you want to know, okay, this beta cell doesn't produce insulin well enough. Can I treat it with 100 different drugs and find one that makes it work better? So that's the whole idea behind using iPS cells for treating patients. This just shows you, and I don't know but this is just a gene profile looking at how similar induced pluripotent stem cells are to the embryonic stem cells. And it's really gotten very, very good. So basically, this is a skin cell that's been induced to be pluripotent. And when you compare the genes that are expressed in that cell, they're very similar to the genes that are expressed in an embryonic stem cell. And this is just getting better and better and more reproducible. And many labs are doing it all over the world. So this is not something that worked once and is never going to work again. It's just the technique is getting better and better. And, well, that's just shown here. So now many different groups can do this. Also, they found out that you don't need all the same factors. Some groups use a cocktail of these 4 factors, and then this has 2 of the factors but 2 other factors. So it looks like our cells have really the potential to be reprogrammed, and that's the whole idea behind induced pluripotent stem cells. And this is just amazing. It's going from right to left, or from your side right to left. Basically, this was nuclear transfer. So everyone was studying nuclear transfer, and really this is just the number of papers that have been since 2006 on nuclear transfer technologies. And this was actually this study was published in about a year ago, very early 2014. But you can see for induced pluripotent cells how much increased in the publications. And that's because it's working and people are really using with it. It's got real potential for regenerative medicine. So this just summarizes what I've hopefully gone through with you, the different types of cell types, or the different types of stem cell populations, and really the advantages and disadvantages of them. And we didn't go into some of this, but basically the embryonic stem cells have probably the most potential to become every cell in the body, but iPS cells are actually very near that. All of these cells now, they still have, if you put them into a person, they still have the potential to become tumors, so that's something we haven't been able to address yet. There are still immune rejection issues. Human ES cells, the problem is that they are derived from human embryos, where the iPS cells can be matched to the patient and become from just a skin cell of the patient. So this is where I think most of the work is going now. People are not using somatic cell nuclear transfer as much, and adult stem cells don't have the same potential. So this is really where the research is going, and for precision medicine, iPS cells are really the root. And so the therapies, basically, like I said, for regenerative medicine and tissue repair, it's getting better, especially for skin cells. You can imagine if you took a person's cells and you could actually grow them in a dish and make a number of cells you could use to treat burn patients. They're using this for eye transplants, so people that have eye disease, they can replace some of the cells in the eye, and that's actually working, and people are regaining their vision. Tissue repair. So it's actually, we're going, in some tissues this is really working, but in other tissues it's not. I think, like I said, I think the real value of this is we can model development in a dish using cells that are normal cells. We can model disease, so we can take a patient's cell that's not functioning properly and figure out why it's not functioning properly. We can use it to do drug therapies. And so, again, drug discovery, I think, is the most potential. So in this patient, you can take their skin, you can reprogram it to be a pluripotent cell, and say you have a disease. Actually, a good example is, say you have a disease or a syndrome, and there's several problems. The person is obese, and the person has diabetes, and the person has cardiac problems. How do you know which is the primary defect? What's the primary cell, and what is maybe a secondary phenotype? Well, if you could take that patient's skin cells, reprogram them into a heart cell, into a neuronal cell, into a liver cell, and say, okay, which one of these cell types is the actual dysfunctional cell? Then you would know the primary cause or the primary place where this disease is gone wrong. So it could be that only the neuron doesn't function properly, or it could be that all 3 cell types don't function properly. So this way you can figure out how to treat that patient, how to treat that defect by knowing which cell type that is actually dysfunctional. So this has been really powerful. Also, there's been a lot of instances where you think it might be primary in this one cell type, and it may not be. And you can tell that by growing that cell in a dish. And again, if you have that information, then you can treat that cell and figure out what drugs could be involved. So this is just a final summary. Basically, we can take cells from an individual, we can reprogram them. If there's a genetic disease, which hopefully precision medicine will identify for us, we can correct that. And we can then differentiate those cells and put them back into a person in the future. Or what we can do is we can basically differentiate them into the cells that have the abnormality and figure out what's happening in that disease and to use it as drug discovery. And if they don't have, at this point, if they don't have it, you could just actually take these healthy cells and maybe give them back to the patient. Especially if they've lost certain cell populations. So this is all a way of taking what we learned from precision medicine, from sequencing these patients, to going into saying where is the actual defect, what's the actual phenotype, and doing this in a cell-based method rather than in a person itself.